everyone, and welcome to another video by BioTeach. For this video, I'm focusing on chemical synapses. This follows on from other videos I've done for the BTEC Applied Science Unit 9, but this is hugely relevant for the A-level biologists amongst you as well. There are a number of videos on a playlist on my channel that are relevant to this unit. If you click on the link that's just flashed up on your screen, you can access them all. So when we're looking at synapses, we're essentially just looking at the junction where two neurons meet. That's what's called a synapse. Neurons never really touch each other in our nervous system. There is actually a very narrow gap between the two ends of the neurons, known as the synaptic cleft, and communication between the two occurs using neurotransmitters, which are basically chemical messengers. A neuron may have synapses which connect with up to 10,000 other neurons, so that's quite a lot of communication that happens. When an action potential reaches the synaptic knob, the impulse of that action potential must be passed on to the other neuron, or it could go to an effector such as a muscle or a gland. So when we look at synapses, we're really just looking at them consisting of some key features. So first of all, we're looking at this presynaptic neuron or the presynaptic knob. Here, we've got the axon terminal, which then opens up into the presynaptic knob. The gap over here is known as a synaptic cleft. And then the next membrane that you see here in the yellow is the membrane on the postsynaptic neuron or the receiving cell. It's the arrival of the action potential in the presynaptic neurons. The action potential travels down this way, and it's the arrival of it which allows these two neurons to communicate. The chemical messengers that I've previously mentioned can come in many forms, but there are two main ones I think you probably need to mention in assignments or you need to know for your exam. Firstly, we've got noradrenaline, which I've mentioned in previous videos when we're looking at how heart rate and breathing are controlled by the autonomic nervous system. The next one is acetylcholine, which actually takes action in synapses known as cholinergic synapses. Acetylcholine is sometimes abbreviated to ACH, so you'll see that in some of the diagrams that I'm going to show you in a second. I've borrowed this animation from dynamicscience.com, and it allows us to see how synaptic transmission actually works. So the yellow star that you see there, that was the action potential. And that basically shows you that coming in from the presynaptic neuron and traveling down into the presynaptic knob. It also shows vesicles that you can see over here, which have a really important part to play in opening up these voltage-gated channels and allowing the neurotransmitters, which are these red blobs, to exit the presynaptic neuron to then reach the receptors in the postsynaptic neuron. So let's just have a look at it in a bit more detail so you can understand the kind of process and what you actually need to write down. So these images that you can see on my next few slides have actually been taken from the CGP A-level book. So I've borrowed them from there because they're nice and clear in allowing you to understand the steps that take place. Now, step number one is when the action potential basically arrives at the presynaptic neuron and moves into the synaptic knob. And this action potential arrival basically stimulates voltage-gated calcium ion channels to open. And we're talking about these ion channels over here. When these open, calcium ions will diffuse into the synaptic knob. Please remember that voltage-gated means that gates will only open once the potential difference reaches a certain voltage. Now, calcium ions will basically diffuse into the synaptic knob. Because these gated ion channels are open, they move into the presynaptic knob. These will be pumped back out later once the action potential has finished, and they're pumped back out via active transport. The influx of these calcium ions moving in basically encourages the synaptic vesicles that are filled with the neurotransmitters, so these blue blobs here. These will basically fuse with the presynaptic membrane and allow the neurotransmitters to be released by exocytosis. Now, the vesicles releasing that neurotransmitter will basically release them into the synaptic cleft. So it's this gap in the middle over here. And as I said, it happens by exocytosis. Exocytosis is just the opposite of endocytosis. It's a process where a vesicle inside of the cell moves to the cell surface membrane, fuses with that membrane, and releases its contents outside of the cell. Now, these neurotransmitters in the gap will basically be complementary to the cholinergic receptors on the postsynaptic membrane. So these neurotransmitters will be in a high concentration at one end, and they will diffuse over the synaptic cleft to land on these receptors on the postsynaptic membrane. Now, this diffusion obviously takes time, and when they have landed on the membranes, it causes sodium ion channels in the postsynaptic membrane to open up. So basically what this means is that the influx of the sodium ions into the postsynaptic membrane causes the depolarization, and that basically means that the action potential will be generated as long as the threshold has been reached. And then the last step that you've got over here is that the acetylcholine will have to be removed from the synaptic cleft. So the advantage of that being removed is basically that the response won't keep happening. And essentially what we've got is an enzyme called acetylcholinesterase, which will break down the ACH and will allow the products of that breakdown to be reabsorbed back into the presynaptic knob. They'll be recycled and repackaged into vesicles for then the next action potential to arrive whenever it does. The nature of synaptic transmission in the nervous system allows what we call the integration of inputs from many sources. Here we're talking about the interpretation and the coordination of these messages. You can actually have excitatory inputs, which basically will cause depolarization of the next neuron, or you can have inhibitory inputs, which basically stops the action potential from being generated in the next neuron. Nerve transmission across chemical synapses has got several advantages despite the delay caused by that diffusion of the neurotransmitter. Chemical synapses will transmit impulses in one direction to a precise location, and because they rely on a limited supply of neurotransmitter, they are subject to fatigue, basically the inability to respond to repeated stimulation. Now, synapses can also act as centers for integration, as I mentioned before, from many sources. So the response of postsynaptic cells is often not strong enough on its own to generate an action potential. However, because the strength of the response is related to the amount of the neurotransmitter released, subthreshold responses can basically sum together to produce a response in the postsynaptic membrane, and this is what we call summation. Now, there's two types of summation. You can get temporal summation, which is shown by this diagram over here. Now, temporal summation is basically where several impulses may arrive at the synapse in quick succession from a single axon. So that's what's this showing over here. This is a single axon over here, and you've got these arrows showing you the action potential that's arriving at the presynaptic neuron. The individual responses, because they're so close in time, they will sum up together to reach the threshold at this end that then releases the neurotransmitter and generates the action potential in the postsynaptic neuron. This graph over here is the action potential graph that you might have seen before. The second type of summation is what we call spatial summation. Now, over here, we're talking about individual responses arriving from separated axon terminals, and they might arrive simultaneously or at the same time at different regions on the same postsynaptic neuron. The responses from the different places will basically sum up, so these two will add together to release neurons into the synaptic cleft to then generate the action potential in the postsynaptic neuron. So I hope that's been super useful for you all. That's a good summary of cholinergic synapses and how neurotransmitters can be released in the synapses to then pass the messages on from one neuron to another. If you've got any questions or anything that you want to input, feel free to leave me comments underneath this video. I have linked some other related videos in the description of this one, so please feel free to scroll down and have a look at those as well. Thank you so much for watching as always. Please don't forget to like, comment and share. Bye for now.